Okay. So we had a question, or maybe we did not discuss much the role of cholesterol in an animal cell, particularly. We see a very clear component of cholesterol in an animal plasma membrane. Then why should we have it? In animal cells, cholesterol molecules are inserted between the lipid bilayer. They are stiff and give stability to the membrane. Okay, so I'm going to show you how a cholesterol molecule fits in the bilipid plasma membrane, giving it more fluidity, giving it more stability, making it more efficient in employing the permeability definitions. If you see this, we have a picture of cholesterol here which says carbon rings. You can clearly see the rings here. It is a steroid. This ring itself is called as sterol ring. This is a carbon ring structure. Then we have a hydroxyl group makes the cholesterol when it is in the plasma membrane. This hydroxyl group will give a hydrophilic entity towards the outside of the plasma membrane. You can see here the OH group, which is shaded red in this picture, is this yellow color head in this picture. So this also plays a role in attributing the hydrophilic character in case of outside the cell or the cytoplasmic end. We can also see I have a hydroxyl group. And non-polar tail attracted to the hydrophobic tails of phospholipids in the center of the membrane. So it has a non-polar tail which is attracted to the phospholipids. So they sit in the gaps. When I have the phosphobilipid layer with the hydrophilic ends outside and hydrophobic ends facing each other, but still there are gaps in between them which can allow the water soluble molecules to seep into the cell or move out of the cell. So this cholesterol will bridge that gap. So it's like I have a brick wall and at places there is a gap between two bricks and you cement it. This cholesterol, it can get attached to the hydrophobic entity of lipid bilayer, also attribute to the hydrophilic entity by means of its hydroxyl group. So it forms a proper incorporation into the plasma membrane. As you are seeing, because of this there is a hydrophilic entity promotion. So that makes it more water soluble, more fluid, and it also gives the structural stability and a definite barrier for permeability when it gets fit into within the gaps of the phosphobilipid layers. These are the functions of cholesterol in plasma membrane.